that is moving to the inside. And then we've got a drag force there that is just opposing any movement. It's a little bit of a teenager. But what happens here is that if we've got a large particle, it will be pushed because it's fed by the centrifugal force to the outer wall, where we've got a flow going down and the particle leaves the system through the underflow. Whereas the light particles, the buoyant force going to the inside is larger because they're not pushed as much to the outside. So the small particles end up in the central flow, in the hydrocyclone, and leave the device through the overflow. So we've got two different liquids coming out of, so we've got a mixture coming in as the feed. The flow swirls around in the hydrocyclone. The fat, heavy particles move to the outside and leave through the bottom. The little particles are sucked to the middle and leave through the top. And that way we can separate different cell types, or we can separate sand and particulate matter from a clean sample to do later analysis with. And this paper is quite interesting because the Manjit Warkiana that realized that we could also do this on the micro scale was kind enough to put all the STL files for 3D printing the hydrocyclone on there as well. So you can, if you get a 3D printing, you can just get the designs, get your printer to make one, and then see how it works on particle separation. Also working on forces happening in a spiral field are these spiral devices, where we've got a sample being injected from a central inlet and spiraling around. Then again, we've got forces pushing the particles to the outer wall, and we've got forces dragging it inside. So we've got Dean forces here going in opposite directions. We've got a flow going, the Dean vortex is going here positive, here it's going negative, and we've got different stable positions for different particle sizes. If we adjust our flow big enough, flow conditions well enough, and that always takes some experimental trial and error, we can actually organize life so that big particles go to the inner wall, small particles go to the outer wall, and we can collect the small and the big particles in different outlets, which is exactly what's shown here. So here we've got exactly the same device on the previous slide, where we've got the inlet and two different outlets, one that collects the flow that has taken the inner curve all the way through, one that's collecting the fluid that's taking the outer curve. If we want to use this as a simple particle concentrator, we work at a high flow rate of 6 milliliters and everything gets swiped to the outer curve. And you can see that all the particles sit here. If we go and use at a moderate flow rate of 2 milliliters a minute, however, the equilibrium of the Dean's forces and the drag forces is as such that the small particles are being pushed towards the outer wall, the big particles are moved towards the inner wall, which means that we can collect particles of different sizes based on the different focusing collection points. So this part of this predictable particle, part of it is trial and error where your inlets and outlets have to be. But again, these devices, you've got a microfluidic structure, you have a pump with a certain fixed flow rate, and without the presence of any membrane, allows you to either exclude all the small particles because they all go through one outlet, or it allows you to separate bacteria from mammalian cells to have a cleaner culture happening. So to summarize, we've got a whole range toolbox full of different techniques that use the different forces. We've got the straight channels where we started off, where we've got the different focus in inertial focusing nodes. We then have the contraction expansion, where we got our small particles to just follow the sides, but because of the pressure points caused by the expansion contraction tracks, the circulated tumor cells were swept to the middle, and we could then collect the big ones from the outside and the small cells from the outside, and the big stiff circulating tumor cells from the inside. We didn't go into any of the serpentine, but if you use complex channel geometries, we also get preferential agglomeration of small particles at at the outside, and the bigger ones on the inside. So there's a range of different structures being made there. There's the spiral devices where there's a couple of different options where with the flow rate you can either put all particles together, have it working in filter mode, but if you tailor the flow rate a little bit lower, you can actually selectively collect one particle type at one wall and the other particle type at the other wall. And then of course there's a whole range of ways that we can mix and match different approaches, active and passive particle separations together using a Lego-like approach. Then we've got a way that we can change the fluids. So that's using a viscoelastic fluid, which the best known one is saliva. So there's a long polymer added, which means that we add viscoelastic effects to our separation system. And in contrast with the focusing node that we had with our normal inertial fluidics, in viscoelastic systems, the particles focus in a spaced manner in the center of the channel. Here we've got a couple of examples of where we can, how viscoelasticity has been used. Here the viscoelasticity is provided by PEO, which is a long chain polymer. Normally with viscoelastic flow, the particles will be concentrating in the middle. But if you use the sawtooth structures, it pushes the force field to the outside. So here we can have the red blood cells that come in from the sample all being pushed by by the pressure implemented by these sawtooth structures. The red blood cells are all pushed to the outer wall, which means that from one outlet you can collect all your red blood cells, and from the other outlet you get a nice clean plasma sample where you can do your chemical analysis on. The same reagent PEO was also used in a very simple structure here to separate, to concentrate bacteria from microalgae. The bacteria were focused in the middle using the viscoelastic effects, whereas the algae were not impacted and pushed to the outside. So we have a system where the minimum energy part for the bacteria is in the center, the minimum energy point for the algae was on the outside, and you use different outlets to separate, fractionate your heterogeneous sample into separate fractions. A last trick is that you can kind of extract large blood cells as well into a viscoelastic medium. So here we've got a normal fluid and a viscoelastic fluid flowing adjacent to each other. The viscoelastic forces drag the large particles in without impacting on the small ones. So that allows you, if you've got a combination of circulating tumor cells and normal blood cells, the circulating tumor cells are larger and stiffer, so they will experience a much stronger pull into the viscoelastic medium than the other smaller blood cells. And that way you can separate two different particles, two different cell types based on their size, using a viscoelastic acceptor phase. Here a viscoelastic microfluidic device was used for the diagnosis of sepsis by focusing the bacteria down into a smaller volume and then making it possible to detect the viscoelastic properties. So here we've got the outlet of the channel. So this is the whole device. We've got the inlet for, where does the sample come from? Yes. So we've got the viscoelastic inlet and the blood inlet coming here together at a first focal point and then going to the outlet. If we look under the microscope here, this is the outlet. So the focused bacteria here we can see in the middle, and they go straight through the central outlet, whereas the clear liquid can be obtained from the outside. We can then do testing of the bacteria in the concentrated sample coming there out of the central channel. There is a couple of other ways of sorting technologies. There is ways of just using a weir-like structure where large particles can't get through, but smaller ones can. Again, if you've got a microchannel and you want to only, you can flow in one direction and allow your small particles to go to another channel, whereas the smaller ones can't. You can use a pillar type structure to pretty much work as a filter. Most of the fluid can get through, but some of your particles will get stuck if they don't fit through the pillars anymore. Cross-flow filtration, as we know from normal filtration, you can also do at a microfluidic scale. You just put a couple of pillars in there, the flow goes perpendicular to the filtration direction. Most of the flow will be carrying along the particles, preventing the filter from getting blocked. But small particles and the particle-free solutes will be leaving the device for the small channel for further processing or detection. Deterministic lateral displacement we already talked about, and there's hydrodynamic effects that we can use as well by squeezing the flow down from the sides. This is the Lego blocks approach where we can combine them together. I think we want to move on to the next lecture. So we've got active and passive methods, where the active methods have the advantage that they provide very precise control, but they typically have low throughput and require extra devices. The passive methods are very simple and high throughput, but they require an understanding of the physics and requires complex channel design, and therefore lack flexibility because you can't change the design while you're doing the experiment. So other than the flow rate, there's not much you can do unless you make and design a new device. And then there's the viscoelasticity, which is very simple, very low cost. It's got good focusing performance, but a lot of the viscoelastic phenomena are not well understood, so it's more so observational, like the sawtooth device that we showed. It is not well understood why all the particles are being pushed to the outer side. So there's still a lot of research to be done to properly understand. And there may be some consequences if cell populations need to be reintroduced into the human body, that the PEO as a polymer may not be the most friendly and may need to be removed after processing. So if you add a polymer, that may potentially compromise the usability of the product. So great if you separate something you want to throw away. If you want to reintroduce it in a biological system, that may become problematic. And then the last slide is a couple of references. So these are the different tools that we have to separate particles of different size, density, and geometry. And again, it's a toolbox. It's not necessarily that there's one is better than the other, but for each application there will be one more suitable than the others. So you'd need to come up with your problem, look at the toolbox, and then pick the right screwdriver. Happy to answer any further questions. And if not, it's time for Professor Malik to talk about solid phase extraction. But we may want to take a short break. I'll leave that up to you.